Hello class. Today we're going to be going over Chapter 3 and cells. The first half of this lecture is going to be talking about the cells themselves and the individual parts of each moving cell. Then we are going to go into active and passive transport and sodium-potassium pump. This might seem a little heavy, but just stay with me. So Chapter 3, please open up your books to cells. In this lecture, I do want to identify and discuss some of the basic cell structures and functions and the major components. We will talk about the functions of the primary cellular organelles. We will compare the major passive and active transport processes that act to move substances through cell membranes. This one is very, very important that you understand. This is what is utilized for our IV therapy. So then we're going to go on and compare and discuss some DNA and RNA, their function and protein synthesis, and discuss the stages of mitosis and explain this importance of cellular reproduction. So let's first talk about just the cell themselves. So size and shape, they can vary considerably in size. All are microscopic. Cells differ notably in shape. As you'll note on this slide, each cell in the body is surrounded by a thin membrane. This is called the plasma membrane. This is a very important thing to understand. This membrane separates the cell contents from the dilute saltwater solution called interstitial fluid, or IF, or simply called tissue fluid, which bathes all of our cells. A small circular body called the nucleus is also on the inside of this cell, and as we go through this, you're going to learn more about how important this nucleus is. So what's the composition? Well, cells contain cytoplasm, organelles, and cell interior. So let's look at some of the parts of the cell, such as the plasma membrane, cytoplasm, nucleus. All of these words are just being thrown around. So what are they? Plasma membrane identifies a cell as being part of one particular individual. Some of its surface proteins serve as a positive identification tag because they occur only in the cells of that individual. A practical application of this fact is made in tissue typing. A procedure performed on an organ from one individual is transplanted into another. Carbohydrate chains attached to the surface of cells often play a role in this identification of cell types. The cytoskeleton is made up of a thin, thread-like filament called microfilaments and tiny hollow tubes called microtubules. The cytoskeleton provides support and movement of cell organelles. So the plasma membrane. This plasma membrane forms the outer boundary of the cell. It's composed of thin, two-layered membrane of phospholipids. Phospholipids is something that we will be discussing quite a bit in your first term of nursing school. Embedded within the plasma membrane are proteins, and it is what we call selectively permeable. It's not permeable, and it doesn't let things in and out, just anything. It is selective in this. So cytoplasm. All cell substance from the nucleus to the plasma membrane. So this is inside that cell. It's just a cytoplasm. Then you've got the cytoskeleton, and that cytoskeleton is the internal framework of the cell. So let's look at some of the organelles that are going to be inside this cell. We have some ribosomes. These are made of two tiny subunits of mostly RNA. Then we have manufactured enzymes and other proteins, often called protein factories. So the ribosomes are these protein factories, and Ribosomes may attach to rough endoplasmic reticulum or lie free in the cytoplasm. So, what is the endoplasmic reticulum, or referred to as ER? Rough ER collects, folds, and transports proteins made by the ribosome. So you can almost think of them as little carriers. The endoplasmic reticulum is a network of connecting sacs and canals that wind back and forth through the cell cytoplasm from the nucleus, almost to the plasma membrane. The tubular passageways or canals in the endoplasmic reticulum carry proteins and other substances through the cytoplasm of the cell from one area to another. Then we also have smooth endoplasmic reticulum. This synthesizes chemicals and it makes new membrane. So moving on to other organelles that we're going to see inside of the cell, we have the Golgi apparatus. This is a group of flattened sacs that lie near the nucleus. The Golgi apparatus collects chemicals into vesicles that move from the smooth ER outward to the plasma membrane. The Golgi apparatus is sometimes called the chemical processing and packaging center. Then we have our mitochondria. These are composed of inner and outer membrane sacs, often called the power plants of the cell. Mitochondria are involved with energy-releasing chemical reactions called cellular respiration, and each mitochondrion contains one DNA molecule. So mitochondria are quite important, these power plants of the cell. Moving on, we have lysosomes. Sometimes we refer to lysosomes as like the digestive bag or the microbe-eating organelle. This is a membrane-enclosed packet that contains digestive enzymes. Lysosomal enzymes can digest and thereby destroy microbes that invade the cell. Then we have our centrosome. This is a microtubule organizing region of the cytoskeleton near the nucleus. The centrosome plays a role in forming and organizing the cell's cytoskeleton, including some of the cell's outward extensions. Centrioles are paired organelles that lie at right angles to each other within the centrosome and function in moving chromosomes during cell reproduction. Then we have these cell extensions. So we have something called microvilli. Microvilli are short extensions of the plasma membrane that increase surface area and produce slight movements that enhance absorption by the cell. Then we have little cilia, and you're going to hear this term again. Cilia are hair-like extensions with inner microtubules found on free or exposed surfaces of all cells. They serve sensory functions, but some are also capable of moving together in a wave-like fashion to propel mucus across a surface. Then we have our flagella. Flagella are single projections, which are much longer than cilia, that act as the tails of sperm cells. So let's talk about this nucleus. The nucleus is the central structure of the cell. It controls cell because it contains most of our genetic code or genome. This is the instructions for making proteins, which in turn determine cell structure and function. Component structures include nuclear envelope, nucleoplasm, nucleolus, and chromatin granules. Within our nucleus, we have 46 nuclear chromosomes containing DNA, which contains the genetic code. So now that we've talked about this, in this image here, you can see how all of these organelles, that plasma membrane, you can see that nucleus in the center, You can see the flagellum, the tail coming off, and you can see how complicated each cell is on a microscopic level. So what are the relationship of cell structure and function? Well, every human cell has a designed function. Some help to maintain the cell, others regulate life processes. Specialized functions of a cell depend on the number and type of organelles. So for example, cells that contain large numbers of mitochondria, such as the heart muscle cells, are capable of sustained hard work. Movement of the flagellum of a sperm cell is another example of the way each type of organelle has a particular function. The sperm's flagellum propels it through the reproductive tract of the female, thus increasing the chances of successful fertilization. So here we come to the most important area of this lecture, and that is the movement of substances through cell membranes. So we have to have some way to move substances from the plasma around the cell, all that fluid, and to get things in and out of each cell. So how do we do that? Well, we have passive transport processes and we have active transport processes. So let's first talk about our passive transport processes. So passive transport does not require added energy and results in movement of down a concentration gradient. So passive transport does not require that added energy. That's interesting. In other words, it goes down from a higher to a lower gradient. We have diffusion, osmosis, dialysis, and filtration. These four make up passive transport processes. So let's look at each one. First, we have diffusion. Diffusion is where substances scatter evenly throughout an available space, and pores must be large enough for particles to pass through. So it's just kind of this, if you want to think of it, it just scatters around and then it can pass through. Then we have osmosis. Osmosis is the diffusion of water when some solutes cannot cross the membrane. So in osmosis, because water moves into a space, but there is no exchange of solutes, a change in fluid pressure may result, called osmotic pressure. Very important concept. Then we have our dialysis, and this is the diffusion of small solute particles. And then we have filtration. Filtration is the movement of water and solutes caused by hydrostatic pressure on one side of the membrane. Filtration is partly responsible for moving water and small solutes from blood into the fluid spaces of the body tissues. Filtration is one of the processes responsible for urine formation in our kidneys. And waste are filtered out of the blood into kidney tubules because of a difference in this hydrostatic pressure. So let's go into this a little further. Diffusion through a semi-permeable membrane. Semi-permeable means that not everything can pass through, right? It's kind of permeable, which means it's kind of opened up to let things through. So let's look at our figure here. At the bottom, you'll notice that we have on the left, we have diffusion, and then we have equilibrium. Equilibrium is that normal scale, right? And if you note in this image, the membrane is permeable to the purple solute particles and to water molecules. And that it separates a 10% solution of purple solute particles from a 20% solution of purple particles. The container on the left shows the two solutions separated by the membrane at the start of diffusion, and the membrane is that going right down the middle. The container on the right shows the result of diffusion over time. Now we have osmosis. Osmosis works a little differently. If you will note, you have our membrane going right down the center. And you'll see where we have this 5% albumin that is going into the 10% albumin, and water is coming out. So osmosis is just a very easy downward gradient. So I want to go into our active transport processes now. Active transport processes occur only in living cells, and the result in substances moving up the concentration gradient. So it's no longer going from a high concentration gradient to a low. In active transport, we're going from a low concentration gradient to a high. Because we're going against that gradient, we're trying to push our way in, it requires energy from something called ATP. ATP is adenosine triphosphate, and this is another extremely important concept to understand. So we have these ion pumps, right? An ion pump is a protein complex that's in a cell membrane. The ion pumps use energy from adenosine triphosphate, or ATP, to move substances across cell membranes against their concentration gradients. So the biggest one that we see here, very, very important, is what we call a sodium-potassium pump. We're going to get into that more in just a moment, but let's talk about the sodium and potassium. So as sodium pumps move sodium ions only, likewise, calcium pumps move calcium ions, and potassium pumps move potassium ions. So you see the relationship between the pump and the actual solute. Some ion pumps work with other carriers so that glucose, which is sugar in our body, or amino acids, which are these building blocks of proteins, are transported along with ions. So, we've got a basic understanding of active and passive transport. Well, then we have some other movements that occur. Let's talk about this. We have phagocytosis. This term literally means cell eating. It engulfs large particles into a vesicle as a protective mechanism. Then it destroys bacteria or debris from tissue damage. Then we've got something called pinocytosis. Pinocytosis means cell drinking. It engulfs fluids or dissolved substances into cells. Okay, so I want to talk more about this sodium-potassium pump. If you look here at this figure 310, it actually shows a sodium-potassium pump. And if you notice the little blue dots on each side of it, that is that plasma membrane that's made up of phospholipids. You can think of phospholipids right now as kind of the slippery little substances. So, let's take a look at what's actually happening here. If you'll note, the pink little dots are sodium. Na plus means sodium. Then the purple that's labeled as K plus means potassium. So potassium is abbreviated as K plus. And if you notice, we have got these little things happening. You can see where that ATP is, and that's that burst of energy. You can see that ADP. So let's kind of talk about this a little bit. The 3 sodium ions on the upper left are pumped out of the cell, and 2 potassium ions are pumped into the cell during one pumping cycle of this carrier molecule. So pictures 1 and 2 go together. As sodium goes in, potassium goes the opposite direction. So if we have a sodium-potassium pump evolving, they work opposite. So if sodium goes in, potassium comes out. If potassium goes in, sodium comes out. ATP is broken down in the process so that energy freed from ATP can be used to pump the ions. Okay, I want to move on, and I want us to talk a little bit about cell growth. This is very important concepts for you to understand. So we have our DNA, which is deoxyribonucleic acid structure. It's a large molecule. It's shaped like a spiral staircase. I'm sure many of you have seen this image. And their base pairings are always the same. They can be thought of as complementary base pairing. But the sequence of base pairs differs in different DNA molecules. So a gene is a specific sequence of base pairs within a DNA molecule. Genes dictate the formation of enzymes and other proteins by ribosomes, thereby indirectly determining a cell's structure and functions. In short, genes determine our heredity. So more on cell growth. So for normal growth and maintenance, the cell must continually produce, and many diverse structural and functional proteins are needed for human life. The functional proteins then synthesize carbohydrates and lipids and help regulate all cell functions. Chromosomes, which are composed largely of DNA, contain the information needed to make all the proteins of the cells. Each DNA molecule is made of many smaller units called nucleotides. Each nucleotide is made up of a sugar, a phosphate, and a base. And remember that each nucleotide is made up of a sugar, a phosphate, and a base. So continuing on cell growth, we also have genetic code. And this is that genetic information. It's stored in this base pair sequences on genes. And then we have our ribonucleic acid. This is our RNA. These are copies of genetic information from the nucleus and carries it to the cytoplasm. We have something called protein synthesis. This occurs in that cytoplasm. Thus, genetic information must pass from the nucleus to the cytoplasm. Process of transferring genetic information must pass from the nucleus to that cytoplasm. Do not forget that. Process of transferring genetic information from nucleus to cytoplasm, where proteins are produced, requires completion of transcription and translation. So more about this protein synthesis and transcription and translation. What are we talking about? Well, protein synthesis transcription is a double-stranded DNA molecule separates to form mRNA, messenger RNA. Translation involves synthesis of proteins in cytoplasm by ribosomes. It's a codon. It's a series of 3 nucleotide bases that act as a code for specific amino acid. The mRNA molecules pass from the nucleus to the cytoplasm, where they direct protein synthesis in ribosomes and endoplasmic reticulum. In transcription, each strand of mRNA duplicates a particular gene or base pair sequence from a segment of DNA. Translation requires use of information contained in the messenger RNA. So let's move on now to cell reproduction. So, cell life cycle, the reproduction of cell, involves division of the nucleus, which is called mitosis, and the cytoplasm. Two daughter cells result from this division. And then we have something called interphase. Interphase is the period when the cell is not actively dividing. It's just kind of hanging out. DNA replication is the process by which half of a DNA molecule becomes a whole molecule, identical to the original DNA molecule. This precedes mitosis, so it's before. Interphase includes the initial growing stages of a newly formed cell, in which a cell is busy with protein synthesis and other growth and maintenance functions. During interphase, and just before mitosis begins, the DNA of each chromosome makes an identical copy of itself. The cell then enters another growth period of interphase before it begins to actively divide. After DNA replication is complete, the cell continues to grow until it is ready for the first phase of mitosis. So, this mitosis enables cells to reproduce their own kind. This makes heredity possible. It is the process in cell division that distributes identical chromosomes, DNA molecules, to each new cell formed when the original cell divides. So, what are our stages here? Well, we have prophase. Prophase is this first stage. Chromosomes, which are these pairs of linked chromatids, appear. Centrioles move away from the nucleus. Nuclear envelope disappears, freeing all this genetic material. And these little spindle fibers appear. In prophase, the chromatin granules become organized. Then we have something that is called metaphase. Metaphase is this second stage. So these chromosomes start to align across the center of the cell. Spindle fibers attach to each chromatid. In metaphase, the nuclear envelope and the nucleolus have disappeared. Then we have anaphase. Anaphase is this third stage. As anaphase begins, the centromeres break apart. Separated chromatids are called chromosomes. Now we're talking about something we've heard before. Chromosomes are pulled to the opposite ends of the cell. Movement of chromosomes occur along spindle fibers towards these centrioles. Cleavage furrow develops at the end of anaphase. Then we have telophase. Telophase is our fourth stage. Nuclei appear in daughter cells. Nuclear envelope and nucleoli appear. Cytoplasm is divided. This is called cytokinesis. Daughter cells become fully functional, and cell division is completed in this fourth stage called telophase. In the adult, mitosis replaces cells that have become less functional with age or have been damaged or destroyed by illness or injury. So this is the process of how we can basically repair our cells, copy them, and this keeps our body going. So what are the results of this cell division? Two identical cells result from the cell division. Growing tissues or replacing old damaged cells. So what's called differentiation? This is the process by which daughter cells can specialize and form different kinds of tissue. Abnormalities of mitotic division can produce benign or malignant neoplasms. Neoplasms are abnormal masses of proliferating cells. So when we're talking about benign or malignant, we're talking about benign as not creating a problem with life. and we're talking about malignant, which oftentimes we describe malignant neoplasms as cancer. So now that we have come to the end of this, you can see that chapter 3 can be quite heavy. I do want you to focus in on what the organelles and the different parts of the cells do. For instance, focus in, which is the power plant of the cell. What is considered cell eating? Look at all these terms. I don't expect you to be labeling the cell in a picture. I want you to understand how the cell works. I also want you to focus in on the active and passive transport. I want you to take a look at the handout that is in this portion of your online learning. I want you to try to understand this tonicity and the osmolarity of fluids. Because in nursing we have something called isotonic, hypotonic, and hypertonic solutions. So I want you to look at that handout and try to understand how, knowing how the cells can let things in and out by the different transport processes, can help you to understand how IV fluids work in our patients. And then finally, please understand the basic concepts of cell division and what they mean. I have uploaded videos for different sections of this lecture. I do recommend that you watch them to help complete the understanding of these concepts. Thank you, and I hope you have a wonderful day.